Good evening, I'm Yvonne, Staff for Science for the Public, and we welcome you to tonight's lecture in our series, our Science Literacy series at MIT First. We'd like to thank MIT's Professor Dan Sitzo for sponsoring and setting up this series, and now the next thing is we're very pleased to introduce tonight's speaker, Peter Girguis, Professor of Organismic and Evolutionary Biology at Harvard University. Dr. Girguis received his Ph.D. from UC Santa Barbara, and he joined the Harvard faculty in 2005. He is one of the pioneers in the discovery and the study of organisms associated with the deep sea ocean vents. And his expertise on these extraordinary life forms, which were a surprise at first, is now being applied in a collaborative project, a large one, to the search for life in oceans on exomoons, such as Enceladus and Europa. Few people have Dr. Girguis's interdisciplinary reach, believe me, which includes microbial ecology, physiology, and biogeochemistry. We will hear how his knowledge of life in the extreme deep sea environment informs the search for life beyond Earth. Investigation of deep sea life is complicated by extreme pressure and darkness of the deep sea, and Dr. Girguis and his colleagues have developed special techniques and instruments for these explorations that are themselves very interesting, and I'm hoping he'll tell us something about the inventor side of this science tonight as well. We are very honored to welcome Peter Girguis.
Thank you, Yvonne, for the very generous introduction. I really appreciate that. And thank you all for being here tonight. It's a little damp and cool outside, and it would be a lot easier to curl up by a fire and read a book. But I hope by the end of our time together tonight, you'll have a sense of how we oceanographers and marine scientists are laying the groundwork for studies of oceans on other planetary bodies. But before we go down that road, let me tell you a little bit, if I may, about the way my lab members and I view the world. We consider ourselves physiologists and biogeochemists studying microbes and animals in the deep sea. So what we're really interested in understanding is, in the perhaps a scholarly way of phrasing it, is we really care about matter and energy flow through the ecosystem. Another way of saying that is all living organisms have to harness life from their environments, and in so doing, they change the environments. Those of us sitting in this room here are breathing oxygen, and we're using it, and we're increasing the amount of carbon dioxide in this room and probably water vapor. And our own human physiological activity really does, change to an extent the conditions in this room. Now, in nature, the way organisms change the environment around them influences the other organisms that live around them. And in my lab, we care a lot about those relationships. Who's doing what when we go to a deep sea environment? What microbe is responsible for making methane or eating methane or chewing up the waste products of the animals that live there? What are the partnerships that exist between microbes and animals? That's a big part of the research in my lab, is thinking about those symbiotic relationships. And finally, how do we do a better job of quantifying the role that living organisms play in shaping their own biosphere? We always think about the biosphere as the place in which we live, right? This beautiful, beautiful habitable Earth of ours is our home, but in turn, human beings, excessive activities aside, all living organisms do shape their biosphere, and that richness of organisms influencing one another and influencing the environment around them, and in turn being influenced by the environment, is really what we care a lot about. So with that sort of preamble, I wanted to then dive in and tell you a little bit more about what we do. Now, after centuries of oceanographic research, I think it tickles me that Arthur C. Clarke summed up my feelings about our planet, right? How inappropriate it is to call it Earth when it's clearly ocean. As a deep sea biologist, I feel compelled to share a few things with you. The deep sea is defined as the ocean that's below 1,000 meters water depth. It's about a kilometer, a half mile down. And that's the limits to which sunlight can penetrate. Below 1,000 meters, there is no sunlight. It is an ocean of perpetual darkness. Except, of course, for the little organisms that make their own light. But it is also a cold place that is not just dark and cold, but the pressures can get immense. For every 10 meters you go underwater, you increase the pressure by 1 atmosphere. In this room, we have roughly 1 atmosphere of pressure on us, right? And so you can imagine at 10 meters water depth, you have 2 atmospheres. At 20 meters, you have 3, and so on. And in the deepest part of our ocean, when you get down, to say the Challenger Deep in the Marianas Trench, you're really talking about pressures close to 8,000 pounds per square inch. Or another way of putting this is if I counted up all the square inches on any one of you in this room, and I said, okay, for each square inch on your body, there's 8,000 pounds, and I just did some simple math, that would give me some large mass, and it's about the weight of 50 jumbo jets. It gives you a sense of the immense pressures that exist in the deepest part of our ocean. Now, the average depth of the deep sea is about 4,000 meters, or 4 kilometers, about 2.5 miles, right? And that is these large, expansive areas called abyssal plains that are really, really food poor because there isn't sunlight down there. They live off of the matter that floats down from above. But in addition to that sort of biological scape, the deep ocean also harbors the world's longest mountain chain. It's the mid-ocean ridge system. If you look at a map of the Earth, it looks like, and you see the mid-ocean ridge, it looks like the seams on a baseball. And it is the 65,000 kilometer long mountain range that harbors anywhere from 75 to 90% of all the volcanic activity on Earth. So that means at least, at minimum, for every 1 volcanic eruption on land, there are 3 that occur underwater. Now, the ocean is a large expanse of water, and it has many, many elements in it, including gold. And one of my favorite trivia questions to ask of you, my dear audience, is if I could get all of the dissolved gold out of the ocean and give gold to each person on Earth, how much gold would each person have? Anybody want to take a guess? An ounce? 10 ounces? A pound? A ton? Tons? What do you guys think? Tons. So we got a guess for tons. Anybody else? Well, it's not tons. It's about 9 pounds of gold for each person on Earth. And that's for an element that's actually exceedingly rare, right? And so, sometimes when I say this, there's the occasional entrepreneurial student in the audience who gets really excited. And I just ask them to remember, we poor scientists when they go out and make their millions. But the deep sea, for we biologists, most importantly, represents 80% of our planet's living space. Think about this for a second. Every other ecosystem you can think of, from deserts to rainforests, tundras, cities, your own home, everything else occupies 20% of our planet's habitable living space. All of them combined. 80% is the deep sea, a world that is completely foreign to us, at least intuitively, right? We don't tend to think of our world as being wet, cold, dark, and under immense pressure, and salty, right? That's not how we think of our world. But that is the predominant part of our planet's biosphere. Now, the ocean also harbors an enormous microbial community. And over recent years, as we began to study these microbes, we began to realize there's about 10 to the 27th microbes in our ocean. That's just sort of some ridiculous number. And that if you were to take that 10 to the 27th microbes and string them out end on end, like so many pearls on a necklace, they would span 10 to the 21st meters. Another big number. Well, what does that mean? That's 105,000 light years. That means if I could take all the microbes in our world's ocean and put them on a string of pearls, put them out, stretch them out like a string of pearls, they would cross our Milky Way. When I first did this, I thought that, you know, they might go from here to, I don't know, Jupiter, or I don't know, the nearest star. So for me, as I've studied marine microbes more and more and start to think about these numbers, I no longer think of big numbers as astronomical numbers. I think of them as microbial numbers, right? So it's a real mind bender. We know there's a lot of them, but we really know so little about what they do. Honest to goodness, even with all the genomic sequencing we've been up to, our understanding of what these microbes are doing in nature is still in its infancy. We know they run our planet. We know through geochemical assessments that half the oxygen you breathe comes from marine microbes. So every breath that you're taking in this room, half the oxygen in that breath is not from trees, it's from marine organisms. The nitrogen on our planet, you know, we all get excited about our protein diets and all that jazz. All that nitrogen comes from microbes that are turning the nitrogen gas in the air, which you cannot use, they're turning it into bioavailable nitrogen, right? That's something that they do that we can't. They interact with the animals and the minerals on our planet, and they really shape our entire biosphere, from the most common living spaces to the very edges of our habitable zones. So what I want to do today is share with you a couple of stories, and I'm going to start with some of our research here in the deep sea, and I want to tell you about how microbes have done something I think rather extraordinary. They act as a link between what happens in the deep subsurface, and I mean in the mantle and in magma. Microbes act as a link between that and what some deep sea animal communities are doing on our ocean floor. And I want to use that to give you an idea about how we go about studying these relationships. And then I'm going to switch gears and tell you a bit about how we ocean scientists are working collaboratively with astrobiologists now to think about how we use these tools to study life that may exist in oceans in our solar system and beyond. So let's start with mantle microbe mollusk. First, allow me to introduce you to hydrothermal vents. How many of you have seen hydrothermal vents on TV or something else? Yeah. They're gorgeous, right? They look like big chimneys that are just blowing black billowing smoke underwater. These were first discovered in 1977, and I would argue they were the single most profound discovery of the 20th century. Completely reshaped the way we think about life on Earth, because we saw communities of organisms living down there that do not directly depend on the sun. And that was a very revolutionary realization, right? That there are microbes that could achieve high densities, and they could care less if the sun was extinguished or not, right? So it's a very different ecosystem than we're used to thinking. Now, of course, the animals that live there still need oxygen like you and me, and that's a product of photosynthesis. But it's those microbes that got us thinking about how these vents might be analogs for life elsewhere, right? So let me tell you a little bit about these vents. So some of you in this room are about my age. Others are a little maybe more seasoned, shall we say. And for some of you, you may be familiar with a coffee percolator, although I kind of gather that this is a hip new thing to bring back the coffee percolator. For those of you who've never heard of or used a coffee percolator, it's basically a pot, you boil some water in it, that water goes shooting up a steel tube, and it comes jetting up as hot water and steam into a basket of coffee grounds and drips through there and gives you this dark, horrible brew, right? But that's my experience with coffee percolators. Now, vents are not that different. Our ocean floor has cracks in it, just like our continent. And those cracks allow seawater to seep into them, and believe it or not, there's actually an aquifer in the ocean crust. It's a weird way to think about it, right? We have aquifers on land, doesn't bother us. But when you think about an aquifer on the seafloor, you know, that seems a bit odd, but it's true. Seawater seeps into these cracks through the crust. And areas of the seafloor where there is higher heat due to molten rock, you know, or magma, or what we might call, you know, a magma chamber, say 10 kilometers down, right? That heats the rock around that, and as the water moves through those cracks, through that hot rock, it gets chemically changed. All the oxygen is stripped out. A lot of the other compounds are stripped out. And it becomes enriched in hydrogen sulfide and metals that dissolve and leach out of the rock. Its pH, or its acidity, goes down, and it becomes this hot acidic water. And you can imagine, as hot water, it is now less dense than the cold water all around it. So it begins to move up through cracks in the seafloor until it escapes the seafloor as a smoker. And that smoke you see isn't smoke at all. It's hot, metal-rich water that cools quickly when it hits the deep ocean water and dumps all of its dissolved metals as little black particles. That's what that smoke is. And that's how hydrothermal vents work. Now, these vent ecosystems, as I mentioned, are dominated by extraordinary life forms. And some of the largest animal communities at vents are animals that are symbiotic with microbes. There's a few different kinds. There's the charismatic tube worms, which you may have seen, in any number of documentaries. And there's some other communities. Lately, we've been studying these snails that are the equivalent of the tube worms, but they live in the Western Pacific, the Southwestern Pacific, which means in the waters off of the Kingdom of Tonga and Fiji and Samoa. Both of these organisms have no gut or a very reduced gut. The tube worm has no gut, no mouth, no gut, no anus, doesn't eat anything. What it does is, as a juvenile, when it has a mouth, is it finds its bacteria somehow, ingests it without killing it somehow, and metamorphoses into this tube worm that's stuck on the seafloor. And then it uses this frilly red part, or its plume, to take up chemicals from the surrounding water to feed its symbionts. So that's what it does. The snails do the same thing, except it harbors those bacteria in its gill. And unlike our tube worm friends here, the snails are mobile. They can cruise around. So I'm going to tell you about these snails. So we started studying these in 2009, and there's two different snails and a mussel that all live down in the South Pacific. In an area called the Eastern Lao Spreading Center. And you don't need to worry about that. I'm just going to call it the Lao Basin. And again, that's the area between Fiji, Samoa, and Tonga. And I'll show it to you on a map in just a moment. But this spiky snail is about the size of somewhere between a golf ball and a softball. So they're big. And they've got these spikes on them that we're still trying to figure out what they do. But as I mentioned, they don't go around grazing on food. They suck up chemicals from the vent water to feed their symbionts. Their symbionts, in turn, do what plants do. What do plants do? Plants use the energy from the sun, and they convert carbon dioxide into sugars, right? We're comfortable with this idea that plants, make sugars, and they use the energy from the sun. Well, in the deep ocean, there is no sunlight, but these symbionts use the energy in the chemicals. They suck up those chemicals, and they harness the energy from chemical reactions, and they convert carbon dioxide into sugars. So these animals, it's like having their own little farm that they grow inside their bodies, right? And they support them by bringing those chemicals into their gills. Now, this is Alvinaconcha. This is Ephemeria. This snail amuses me to no end because we go to great lengths to keep all these things alive. And on occasion, if some of our high-pressure aquaria fail or something goes wrong, we'll lose some animals and we'll feel bad, but we don't actually know how to kill Ephemeria. It's a little eerie. Like, I worry that maybe if we release it on land, it's going to take it all over, which of course is a joke. But these guys are tough. And these mussels live down at hydrothermal vents as well. I'm going to focus on this snail, Alvinaconcha, for the rest of the lecture here. We went out and we wanted to ask a simple question. At least it started off as a simple question. We said, we want to know if, and this is the Lao Basin here, this is a 300-kilometer stretch of hydrothermal vents, and there are 4 different vent fields, names Kilamoana, Tokam, Abe, and Tuimulila. If the snails living in here, right, had symbionts that were using sulfide, which is what we have long believed all of these symbionts do, how in the world do they compete for it? Like, who wins? If you've got Alvinaconcha there, and Ephemeria, and this mussel, and all of their symbionts use sulfide, you would think that over evolutionary time, one of them would get better at doing it than the others, and there would be some degree of competition, or maybe they're not competing. Like, this is a very fundamental question for ecologists, and we frankly didn't have the answer, okay? All we knew when we started this is that these snails have a certain kind of symbiont called, I'm just going to give you a shorthand name, gamma A. That's all you need to know. It's gamma A. It's a certain kind of gamma proteobacteria. That they had gamma A, and that gamma A uses sulfide and oxygen, and they take up carbon dioxide and make sugars, right? So the snail, Alvinaconcha, with its gamma A symbiont, cruises around, looks for sulfide, runs it over its gills, symbionts take it up, harness the energy from the reaction, and produce sugars from the carbon dioxide they also take up, and everyone's happy. That's what we knew. But we were trying to get at this question, well, is Alvinaconcha better at this than Ephemeria and so on? But before we started asking about those relationships among these species, we just first wanted to make sure that all of our Alvinaconcha at these different vent fields were doing what we thought they were doing. Now, this work was led by a student named Roxy Beinart, who's formerly a grad student in my lab. She's now a professor at the University of Rhode Island. And she very wisely said, Peter, if we're going to go out there and we're going to collect 288 snails, let's just make sure that they all have the symbiont we think they do. Okay. Because she noticed in the Indian Ocean there were some of these Alvinaconcha with a different kind of symbiont. All right, right? Simple enough. Now here's what we find. We take all 288 samples and we start extracting, we bring them on board ship, we start extracting DNA to look at the host and the symbionts. And while we were collecting them, just so you know, we were making all sorts of chemical measurements around them and grabbing rock samples and trying to get the context. What's the context? Where are these snails from? What's the temperature they're living in? What's the chemistry they're experiencing? So we take these 288 snails, and we've got all this, geochemical context now, and we start on board ship doing this thing called PCR, which is a way of amplifying DNA and using it for sequencing. We were taking a shorthand approach where we could do PCR with very specific chemicals, and it would tell us if it was a gamma A symbionts, right? A screening tool. Think of it as like DNA fingerprinting. Not so different from that. And Roxy wisely said, well, let me make some screens for the other kinds of symbionts that are found elsewhere. Okay? And sure enough, on this cruise of the 288 snails, we found a bunch of them that had 3 different symbionts, not just 1, 3. Oh, we're like, okay, wow. In one place, we have one species of snail that has 3 different kinds of symbiont. That's already cool. How does that work? Which symbiont wins? Do they live together, right? We didn't have all that data. We're still on board ship, and we're out there trying not to throw up, and at the same time being super excited by mama humpback whales with their babies, swimming out in the water, because Tonga is where they breed. So you can imagine a lot's going on, but we get these data, and we're like, okay, well, let's keep going with our work and see where this leads us, right? We also found, once we came back home, that there's not one, there's 3 species of snail. And then at this point, we thought things got easy. 3 symbionts, 3 species of snail, slam dunk. They form pairs, and we're done. And for evolutionary biologists, this is really interesting. Like, you've got 3 different, species. They're in the same genus. They're all Alvinaconcha, but they're 3 different species. How did they figure out which symbiont they want, and so on and so forth, right? When we started looking at the distribution of where these 3 different species of snail were found, it looks something like this. The bigger the white blob means there's more of them at that vent site. So let me just be clear here. At the site called Killamoana, right, we found a lot of this snail, Bouchetti, we'll call it Bouchetti, and some of this one, the Kojamai. And at Tokam, we found a lot of Bouchetti and some Kojamai. When we went down to Abe, I don't see as many. We see a lot of Kojamai, and Strumeri starts to show up. And then when we go down to Tui Malila, there's no Bouchetti, Kojamai's there, and there's a wallop load of Strumeri. So let's look at it this way, just to squint our eyes a little bit, but down here, you got a lot of Strumeri. Up here, you got a lot of Bouchetti, right? And Kojamai sits in the middle. You can kind of break it up that way. Bouchetti, Kojamai, Strumeri. All right, well, what drives that? And our first thought was, at vents, maybe there's no way for these larvae to get from one vent to the other, right? And so maybe they're just spatially separated. But our own data refutes that, right? Our own data shows us here that Kojima is everywhere, right? So it's not likely that they can't get around. All right. Now bear with me, because this is where it gets a little weird and a little complicated, and then I hope you'll begin to see how this all ties together. So then we started to ask ourselves, which symbiont partners with which snail? Right? By the way, at this point in the research, I'm not sure I cared, because I was getting dizzy, and Roxy and I were like, well, you know, game over. This is just bizarre, because watch what happens. This one, we'll call it symbiont E, and then we got symbiont gamma A, and then another one symbiont gamma B. Those are the three symbiont types. And then we've got Buschetti, Kojima, and Strumeri, whom you're all old friends with and familiar with. This is what we saw at first. We're like, okay, so far so good. That's easy. That's not the only relationships we found. We found that sometimes E likes to live in the gills of Kojima, right? And that sometimes gamma A likes to live in the gills of Strumeri. And so now the question is, okay, how does Kojima pick between these two? What influences that? Right? Now remember, you have this symbiont in you. It's living in your gills, right? And if you're getting something beneficial from that symbiont, you want to keep it around. But if it's a deadbeat, you probably want to get rid of it. And there's lots of evidence for this in nature. So we're trying to figure out, how does this work? And then of course, Strumeri can partner with two of these. But then it gets even weirder, because some Strumeri actually seem to have a steady balance of the two of those. And I'm going to tell you in advance, we still don't understand this steady balance. Okay. Now, let me see if I can clear this all up. At this point, we were, Roxy and I were beating our heads trying to figure out, well, what is this all about? We've got certain kinds of snails living up here, and certain kinds down here, and certain kinds here, and they partner with different kinds of symbionts. And what does all this mean? It's not the barriers to dispersal. We ruled that out. It's not that the snails have a physiological tolerance, like, Strumeri is much better at dealing with something down here. The vents aren't that different, okay? But they are different in one major way. The vents at the north, if you look at the rocks are called basalt. If you look at the vents at the south, the rocks are called andesite. And andesite is a silica-rich mineral. And you don't need to know anything more than that, other than the fact that they're different. And when you heat these up and you pump water through them, the chemical reactions that take place between the water and the rock are different. And the basalt, in the presence of the water, makes a lot of hydrogen gas. Remember that, because that is the secret to this whole story. When we took our geochemical sensors and we went down and we looked at the differences in hydrogen concentrations from north to south, there's a wallop load more hydrogen up in the north. And hydrogen for microbes, by the way, that's like a delicacy, because hydrogen is so energy rich, right? We know that. Everybody invokes, oh, look what happened to the Hindenburg, right? I mean, hydrogen's got a lot of energy in it. So these vents naturally make hydrogen gas. And I want to be clear here, some microbes can use that hydrogen, not all of them, but some of them can. They can harness the energy from hydrogen. And for the microbes that can do that, hydrogen in the environment is just gangbusters. That's what you want, okay? So, as it turns out, when we look at Buschetti, this snail, anytime it has an E or an epsilon symbiont, it's found in highest abundance here, where the hydrogen is highest. And these snails, they're gammas. These gammas cannot use hydrogen. They can only use sulfide. And so the whole secret to this pattern here is that the vent chemistry enables certain snail symbiont combinations to do very well, right? So if you're a snail and you can have a symbiont, the epsilon kind, and it can use hydrogen, you're going to get a wallop load of energy from your symbiont if you live up here in Killamoana or Tokam, because there's plenty of hydrogen dissolved in the water. Down here, there's not a lot, and the free-living microbes eat it, and it gets oxidized like it's slippery hydrogen. So you need a lot of it to be around for it to be advantageous. But that's precisely what happens, is that when we look at the hydrogen concentrations, they're much higher up north than they are down south. Now, what does all this mean? Here's the scientific babble for what this means. The patterns we see imply habitat partitioning by species according to the specificity for symbiont type. So let me turn that into English, because I barely understand this. What this means is that if you are an Alvinaconcha snail and you have congeners, that means other species in your genus. Think of them as cousins, right? Competition within the same genus is something that ecologists think about all the time, and if there's a way to alleviate it, that's a great thing. And what we see here is that some of these Alvinaconcha seem to be really good at partnering with these epsilons, and they can't partner with the gammas very well at all. They're predominantly partnering with the E-type or epsilon symbionts, and they live in the northernmost vents where there's enough hydrogen from the water-rock reactions. In the south, where there isn't enough hydrogen, some of these other snails partner with different kinds of symbionts, the gamma A and the gamma B. And they live in areas where sulfide is present, and there isn't enough hydrogen. Now, I'm not going to belabor you with the differences between gamma A and gamma B, but even they show their differences. They use different flavors of that hydrogen sulfide, so even they don't compete with one another. Now, I'm sitting here all excited about this, and you're probably wondering, why is this a big deal? Well, I'll give you the punchline now. I'll show you a little bit more data, and then we'll come back to the punchline. The punchline is this. This is one of the few times where we have shown that a symbiont can influence the distribution of an animal based on what that symbiont can do. And to add to it, this is the only time we've shown that the symbiont is being influenced by abiotic geological processes that are taking place beyond the life. No, there's no life down in the magma, but you've got this really hot magma melt creating where you have these different seafloor compositions because of plate tectonics that create differences in the fluid chemistry that influence the ecology of these little snails. So the mantle, processes in the mantle are influencing what this mollusk is doing. That's crazy. But if anything, it underscores the relationships here on Earth that exist among living organisms and our planet's biosphere, even the mantle, right? So that's what got us excited about this. And it's this really elegant story of how these microbes stitch everything together, because they're the ones who can use the gases that come out of this water that's produced on the deep subsurface where basalt and andesite interact with hot water. And they're the ones who can use the hydrogen or the sulfide, and they're the ones that partner with the snails. So that's the story here. But I want to share one little fun other aspect of this of our work, and that is showing you that they're an epsilon or a gamma kind of symbiont doesn't actually prove to us that they're making the proteins and enzymes that do that work. So this is one of the most ridiculous things we have invented in our lab. The idea was we wanted to be able to preserve the proteins on the seafloor. And the genes that they produce, right? The gene copies, the so-called message RNA. And if I take that snail and I throw it on my submarine and bring it to the surface, and I get the proteins of the genes out on board ship, you know what those proteins and genes tell me? They tell me that that was a crappy ride from the seafloor to the ship. That's what they're going to tell me. So if I extract them, I'm looking at a snail that's under a lot of duress and really unhappy. It doesn't tell me what the conditions are like on the seafloor. So we thought, we have to blend them. We've got to blend these underwater, an underwater blender. And so this is in fact what you're looking at. We used hydraulic motors, which we hooked up to the submarine, and they spun these little blender blades around really fast, and we could take this lid off, which is magnetically latched, and drop a snail in there and blend it. Now I want to be clear, we're not doing this to be evil, or masochistic or whatever it may be, I guess not masochistic, we're not doing it to be evil, but we are masochistic. That's a whole different conversation, a whole different lecture. But what we are doing is trying to get at that pattern of gene expression. So I'm just going to show you a fun video to give you a sense of how this worked, but also how hard this is to do underwater. These are giant robot arms that are incredibly clumsy. I mean, it's amazing what we can do, right? But look how clumsy this is, So we drop the snail in there, the lid goes on, it gets magnetically latched, and then we blend, right? So this is how we look at these patterns of gene expression. And in the interest of time here, because I want to definitely tell you about our other work, essentially what we got out of it was what we had initially hypothesized. We found that the proteins being made by the epsilon symbionts are using hydrogen. So that explains why any snails with an epsilon symbiont are most abundant up here. They're doing fantastic because there's plenty of hydrogen. And down here, these two different gammas are using the hydrogen sulfide, also in the vent fluids, right? But there's not a lot of hydrogen, so they do great down here, right? And they use hydrogen sulfide a little bit differently from one another, but it's really cool. So that little blender approach allowed us to literally, corroborate and demonstrate what we had hypothesized from the earlier data. So this is what I was getting at. This is this mantle to microbe to mollusk, right? This idea that processes that happen in the magma chamber, you've got a magma diaper here, we've got these shallow magma chambers. By shallow, I mean, maybe 10 kilometers. And that seawater moves through here and gets cooked under here. And depending on the composition of the crust, the resulting gases end up being different, and that influences the symbiont, which influences the snails. That's pretty wild, right? And that's why we're so excited. Now, this is how we try and understand the relationships between rocks and water and animals and microbes on Earth. And most of you are here because you probably want to hear about this, right? There's a lot of interest in extending the search for life beyond Earth. And there are a lot of bodies in our solar system that actually have water. Oceans that are bigger than ours. Our ocean's kind of small, that medium, right, compared to other oceans. From Enceladus to Europa, to Callisto to Titan and Triton and Ganymede, all of these have oceans of one kind or another. And astrobiologists and oceanographers are now working together to try and figure out how do you search for life there. Here on Earth, we've come up with these kinds of definitions. I think many of you have heard these before. You probably came up with them on your own in many ways, right? Living things need to take energy. They get rid of waste. They grow and develop. They can respond to their environment. They can reproduce. And they evolve. And this is a great, reasonable list of how you might describe life, right? But how do you test this beyond Earth? Like on Earth, I can take something and put it in an aquarium and see if I get 2 of them. Guppies, for example, as my kids will tell you, right? 2 guppies have now turned into a ridiculous number of guppies. And that's consistent with them being alive. But if we're going to go study life on another planet or another moon, we're not necessarily going to be bringing it back and trying to grow it here for a whole bunch of reasons. People are very cautious about contamination in both directions. Not that we assume that they're going to be evil microbes, but we just don't know what's going to happen. So we want to go and be able to study life without bringing samples back and doing the things we normally do. Astrobiologists have basically used statistical models to ask themselves, and simulations to ask, well, what's the probability of life, right? So here's sort of what's called a Bayesian framework for exoplanet life detection. This is the possibility that there are other planets out there orbiting other suns, and they can, there's a whole bunch of different variables here. What's the distribution of the biochemical states? These are the sorts, this is the posterior sort of probability. What's the frequency of biological innovation? And all these different attributes, and this is fine, but it's a model. And so where we're headed now as a community of oceanographers and astrobiologists is to say, are there more agnostic chemical biosignatures for life? Let's go and see if we can find a fingerprint. Rather than make a statistical guess and rather than try and keep something alive, is there something that's diagnostic of life? Now let me give you an example. Some colleagues of mine have been thinking about the complexity of chemicals. At what point, when you look at chemical complexities, you start stringing molecules together. At what point do you go from biological or from abiotic to something that life has to make? So let me give you an example. Glucose, right? You got 6 carbons that form a ring and some hydrogens and oxygens, all right? Well, yeah, that can be formed abiotically. That's not a problem. We have things like, octanol or decanol or so on. And so any number of these alcohols can also be formed abiotically. But what's really interesting is when you get to a molecule that has 15, a string of 15 carbons or 15, any other elements, then there's very little evidence for that occurring abiotically. Does that make sense? so that there's a certain degree of chemical complexity that seems to be diagnostic of life. And so this is one approach. Let's go and let's just look at how complex some random chemicals are. And here on Earth, when you do it, it works extraordinarily well, right? So I can take something that was alive, you know, like a can of peas, and still see a lot of complex molecules. And I can go out into nature and I can see biology's chemical fingerprint. There are other things that in my lab in particular we've realized. We've learned that microbes produce their own electrochemical signals. If I take an electrode, right, let's say a graphite rod, and I go stick it out in some mud, and I start putting a potential on it, right, an electrical potential, right, or sending let's look at it this way. Let's say I send a current down it. If an organism's interacting with that, it has a very particular set of attributes. So if I do this on soil that's been sterilized, it actually looks different than soil that has live microbes. That's pretty cool. And I haven't grown or cultured or tried to do anything. All I've done is gone and I've sent an electrical charge into the soil. So this is where we're at in trying to think of how do we measure these chemical signatures. And so there's a whole suite of tools, from mass spectrometers to isotope analyzers to these so-called bioelectrochemical systems that we work in my lab. Some people call them microbial fuel cells when they use them to try to harness energy from microbes. Same concept. But a whole bunch of different tools that allow us to ask these questions and look for these agnostic chemical biosignatures, right? That really are agnostic. By agnostic, I mean they don't need a cell to divide. They're just going to tell you if there's something potentially living there or not. We also have a bunch of new tools in ocean sciences that are allowing us to study our ocean without or with little human intervention. And I wanted to just point some of these out to you. This is a device called the Sail drone. It's a solar-powered drone that floats on the ocean. It's amazing. And it just sort of runs and collects data. AUVs, or autonomous underwater vehicles, these torpedo-like devices skim through the water and collect water samples and do all sorts of crazy, interesting things, right? This is a remotely operated vehicle, or ROV. This is a larger machine that's controlled by a human, but it's controlled through a fiber that's a little thicker than a human hair. And that means this makes this thing a lot more nimble. Our older versions of this had a big old cable that was like, keeping your dog on a leash. This makes them a lot quicker and more brisk. And then even these autonomous, think of these as like the Internet of Things for the ocean. They're floats that we put out there that collect a bunch of data, and they come to the surface, beam it to a satellite, and it comes home. That's where we're at. And in my lab in particular, though, we're taking or tackling a very specific task, and this is what I'm going to share with you for the rest of the talk. We're really interested in these chemical signatures again, the chemical signatures of life. Here on Earth, we know we know there's life in the deep sea, but what I really care about is understanding how are these organisms impacting the chemical regime, or how is the changing chemical regime due to human activity impacting these organisms. So change happens both naturally and thanks to our meddling. But in either case, we want to be able to really understand what's changing. And that means making chemical measurements. There are a whole bunch of different ways to do this. People go out with a ship and an ROV or a submersible and they do this. That costs $50,000 a day. Let's put it in perspective. We've built cabled observatories where we run cables down and we power sensors, and that's cool, and they work great. But you're stuck at one point. So I've got kilowatts of power. I can throw all sorts of crazy instruments on there. And it's a great test bed, but I have to sit there. I'm not going to go and wander off. So in my lab, we've taken on the task of saying, can we build a mobile, autonomous, self-sustaining observatory that we can put wherever we want and let it do its thing? And so we came up with this. This is our autonomous biogeochemical in situ sensor system, or the ABIS. I'm terrible at acronyms, so I'm really proud because I finally got one that sounds okay. But this is something that NASA funded because they wanted me and my lab group to really focus on thinking about how you make these chemical measurements in a suite of chemical sensors. How do you link those data? So if I'm going to stick an electrode in sediment and make a measurement, maybe I can reach down there and take a sample for my mass spec as well. Or how do I preserve some in the case of our ocean, to bring back to study the microbes. Or if I'm doing this on Enceladus, something similar in concept anyway, what sensors work best together to give us a diagnostic fingerprint of life? Not one of these is going to tell you that there's life for sure, period. Not one. So you got to put them together and have them give you a repertoire of data. So the abyss is cool, in my opinion, if I may say so, because what we've tried to do is to not reinvent the wheel. So I am not a card-carrying, formally trained engineer, and I will admit to you that this has been an incredibly sort of powerful tool, kind of knowing I'm not an engineer, right? But also not being afraid to try something ridiculous in the lab and knowing when to turn to engineers for help. And so for those of you who have young kids who may be getting a car, I would say do what my parents did, right? Buy them a crummy car. Right? What I learned from having to keep my car alive so I could go to any, the football games and the pizza parties and all that, taught me a lot about mechanical engineering and electrical engineering and so on. Right? So I'm being a little cheeky. But I'm saying sometimes we as biologists or chemists or scientists, we may think, oh, we're not engineers, we can't do it. That's nonsense. What's been great is that we saw these problems that we wanted to tackle. And when we looked out into the commercial marketplace, we realized that a lot of people had solutions to what we wanted to do. And our challenge was figuring out, how do I take this commercial off-the-shelf sensor and make it work at 2 tons per square inch pressure in pitch darkness with a bunch of corrosive salt water around it, right? And that's actually a much less daunting challenge than trying to build your own mass spec, starting with the circuit boards. So a lot of what's happened in my lab is we've built underwater mass specs that we design in-house and are open access to the community. Anybody can build them. We throw out the plans. You want to build one? Go for it. We have underwater isotope analyzers. That's another neat way of getting a biological fingerprint. And all sorts of other underwater sensors and samplers. And we've integrated many of them onto this about 5 foot by 5 foot platform that has this mast and a bunch of flotation on the top. So you can imagine, think of this as sort of analogous to a hot air balloon. It provides flotation even in the deep, deep sea. We drop this thing over the side of a ship, and all these chemical sensors are programmed to do what they want to do what they need to do. And they get to the bottom and they turn on and they run. Now people have been doing that kind of instrumentation, albeit maybe not as sophisticated, for the better part of the last 30 years. What's different here is we thought, how can we do this and give us a degree of control and interaction that we may not have had before? Communicating underwater is a real bear, right? Because you can't just use Wi-Fi. And so my colleagues at Woods Hole Oceanographic who are on this project have developed this thing called an optical modem, which allows us to use flashing lights to communicate at broadband speeds underwater. So now our chemical observatory can be sent to the seafloor, and using this device here, it can flash a light on the top, and then from our ship, we can lower the other flashy light on a cable and get to within 100 meters and talk at broadband speeds wirelessly, just like underwater Wi-Fi. I cannot tell you how cool this is. And that's like the secret sauce to this whole project, because it means that I can put all these instruments on there and cameras, and soon, next year, we're going to be putting on this manipulator, a little rudimentary looking here, but this will actually allow us to turn the cameras on and move the manipulator and watch the video in real time from a ship without physically touching this. That is so cool. And that's what really NASA wants us to do, is not just the technology, but then to ask, when is the right time for humans to intervene? When is it best for people to get in there and make decisions? Because robots and autonomy and machine learning are great, but I'll tell you folks, there are some things that people do very quickly, way better than machines, right? And it's not crunching numbers in Excel spreadsheets. It's a decision-making point, and that's also part of what we're trying to do through this project. So I want to show you this little video, and I want to show you one of the coolest things. Here we are lowering the abyss this past summer. This is off the coast of Southern California in 2017. And we just, released the hook, and it sinks on its own because it has some weights. When it gets near the bottom, it releases those. Here we are with a robot sub. I'm sorry, it's a little hard to see. And do you see that little blue light there? That is the optical modem turning on. Now here's where it gets crazy. I'm afraid that let me just narrate this for the sake of the video, but this is actually a selfie of the ROV that has the optical transducer on it looking at our Abyss lander. So let me back up. The Abyss lander has cameras on it. It's taking a picture and it's sending it through the optical modem to the ROV, and we end up looking at that image of ourselves on the ship. Like, it's pretty wild. And here we are, flashing and talking to one another through these strobing LEDs. And I'm sorry that it was a little hard to see, but afterwards, if we all want to turn the lights off, I can play it again and you can get a little bit of a better scene. But we did this deployment and we had an ROV with us that allowed us to do the testing that we needed and so on. But in the future, we don't need an ROV there. All we need is a ship with one of these flashy, lights, as I call them, and they just talk to this lander. Or better yet, we send an AUV down, a robot sub that goes down, sets up an optical link, collects the data, brings it home, we look at it, and we think, oh, cool, let's change the geochemical sensor profiling and let's do something different. Right now we're releasing the weights here, and you'll see that it's on its way home. It lifts up the bottom and it comes back home, right? So this is, this is like NASA moon landing stuff right off of Los Angeles, where I grew up. And it was a fantastic demonstration of how we can bring together these tools for ocean sciences and use them to set the stage for how we might study oceans on another planet. So this work, and the work of, frankly, many other oceanographers, really aims to address these key questions. Where can organisms thrive, and what are they doing, and how many of them are there? And so this is really the first time in the history of ocean sciences and astrobiology that we're really deeply working together. There have always been the occasional kind of radical, disgruntled ocean scientist who jumps ship and goes to astrobiology, or vice versa. But now the two communities are really putting their heads together to solve some problems. And as I mentioned, here's why. If you look at the size of the ocean, if you took all of the water on Earth and you turned it into a sphere, it would be about that size. And if you look at the volume of ocean on other moons, and Triton and Titan and Ganymede, I mean, it's huge. So this table alone gives you a sense of just how more expansive the oceans are on some of the other bodies. I mean, Europa has an ocean that's larger than our own, and Europa is a small moon, right? So we're going to be faced with the challenge of exploring a giant ocean and looking for these chemical signatures. And I think the chemical signatures are one challenge, but I think in wrapping this up, the biggest challenge is this. We're trying to study life over tremendous spatial scales. Microbes are a micron or so in size. Our oceans are immense. Our planets are spread across light years. We're talking 19 orders of magnitude or more. And for any of you studying a process, to go like 1 order of magnitude is a lot, but 19? And that's one of our biggest challenges. And so I'll end by just showing you some of the kind of open design tools that we've developed in the lab, and our hope is that oceanographers around the world will adopt these for use in their labs and for their own science and science projects. It's free of charge. We've been supported by NASA and NSF and NOAA to build these tools. And as far as I'm concerned, you taxpayers footed the bill, they're your tools. And this open design policy has been really fruitful in fostering a lot of collaboration, right, with people who have ideas on how to improve our tools. So I hope gone are the days of scientists kind of hoarding their own little widget in their lab, because there's way too much work to do. And I've just never seen the benefit of it. But I hope that these kinds of tools, from our mass spec here to the isotope analyzer to our silly little blenders, even to just these incredible little temperature sensors that we can build for about $100 that can withstand full ocean pressure. There's a lot of cool things that we can do. And I mentioned microbes earlier. We can even build systems now, which we've done recently, to harness the electricity from microbes living in mud. They'll make electricity. If you give them the chance to put it somewhere, they'll make electrical current, and you can harness it. And we used it to set up the world's first microbial-powered, quote-unquote, wireless observatory. And we're not powering optical modems. We're sending data acoustically, which is like old dial-up modem speeds. But it was all powered by the microbes. So think about that for a second. You're harnessing microbes to generate electricity to run sensors to study microbes. I think it's pretty cool. But with that, I'd really like to thank you for your time, and I'd be happy to answer any questions you may have. So thanks again.